All right. Well, thanks for having me this morning to talk to y'all. Like Wilma said, I started mid-August at CSU. So been on the job about 3 months, working. I came in during kind of the transition between harvest being done and getting ready to plant the next season. And so that's kind of when I came in, probably the busiest time of the year. And so I just figured out how to fit in however I could. And then me and Scott have been working on the transition ever since then. And still I'll kind of continue through the end of the year until he fully retires in January. So just the outline for my presentation, growing from success. So I'm just going to talk a little bit about previous research and collaboration that I had going back to the University of Arkansas, where I was a soft wheat breeder for the last 10 years. And then talk about stripe rust, which is a problem here. It's also a problem in the eastern part of the US. More or less a problem in all wheat production areas. I'm going to talk about some work we did on stripe rust that's also pretty relevant to Colorado. And then on Fusarium head blight, which is not a problem for Colorado, thankfully. But a little bit of information there because it was a big focus of my research prior to coming. Plowing into the future, just a little background on the CSU wheat breeding program. And then I'm going to just talk about a couple of things. We recently released a new variety for the coaxium production system. So I'm going to talk about that system a little bit. I'm going to talk about wheat stem sawfly, and then a little bit about some drone work that I hope to continue in the future here at CSU. So if you have questions, yeah, you can chat them in. Wilma, if you see those pop up, if you want to interrupt me and ask a question, I'm perfectly fine with that too. There is a hand raising thing on Zoom, but I don't know if I'll be able to see it because I can't see everybody. But however you want to do it. So US, wheat production, thinking about that, right? We think about wheat makes bread, makes, doughs for pizza and donut. I had some donuts this morning. It's actually my 11-year-old just turned 12. So it's his birthday this morning or today. We had some donuts this morning. But, wheat's not actually just wheat. There's 6 classes that are grown in the US. And I'm going to see if I can turn there we go, turn that into a little laser. Right, there's 6 classes of wheat grown throughout the US. In my previous life, I was targeting this eastern US region, which is primarily soft red winter wheat with like a little pocket of soft white wheat up here in Michigan. I mainly targeted this area, eastern Arkansas, down to Louisiana, Mississippi, over to Georgia, South Carolina, North Carolina. That was kind of my territory while I was at the University of Arkansas. So it's smaller wheat production. It's mainly a soybean, corn, and in these kind of Gulf states, there's quite a bit of rice production. Arkansas produces about half of the rice production in the U.S., which is about 1 million acres in Arkansas and about 2 million total in the U.S. So that was my previous area. And of course, we're here in this hard red winter wheat growing area. So this is going to be your higher protein bread wheat production area, much larger acreage throughout this Great Plains region of the U.S. While at Arkansas, I was part of this breeding cooperative, and we worked on as a collaboration to keep all of these programs productive. And so it was called SunGrain. We had the slogan, partnering to predict. And so it involved Arkansas, Texas A&M, Louisiana State, University of Florida, Georgia, Clemson, as well as North Carolina State. So we had this coordinated field testing and, as a group released 50 plus cultivars over the last 10 plus years. So it was a very, very fruitful collaboration that I learned a lot from. I learned how to work with other breeders and and really learned how to develop and release varieties in this collaboration. And one thing that
I don't know if you gotta watch the whole thing or have it on until I give you the code.
one thing we worked a lot on was stripe rust. This is a significant constraint to wheat production, soft wheat production, hard wheat production. Stripe rust is an obligate parasite, and so it needs the host for its survival. It needs wheat or some secondary host for its survival. And one thing about it is it can appear early in the season and it can spread very rapidly when conditions are favorable. And in the eastern part of the U.S., where you get a lot of moisture, a lot of pretty average cool temperatures during the wheat season, you get a decent amount of wind to move spores around, it becomes a serious problem. Cost for aerial application, which is what was mainly used, $10 to $16 per acre. This is a significant cost to wheat production. We know the economics of wheat production are very, very tight. And so every dollar is important. And so we worked a lot on this. And you think about, if you want to combat a disease, you need resistance. And unfortunately, a lot of the resistant genes that we use or have used have really become ineffective. And so this table here, or this graph here, has different resistance genes on the x-axis. And on the y-axis, it has the frequency of surveyed stripe rust isolates that are virulent on that resistance gene. And so most of the resistance genes present in wheat, there are isolates of stripe rust that can attack and defeat those genes. And so in soft wheat, YR17 is the most present gene. Same thing in hard wheat in the CSU program. It's present at a very high frequency. So by itself, it's not necessarily effective. And then YR9 is another one for us, and that is pretty well defeated. And so the goal, when I came to Arkansas, was to identify new sources of resistance, bring those sources in, select and breed for those new sources of resistance. I'll just mention YR5 and YR15 here. Scott Haley, prior to me coming, has done a good job of bringing in these two resistance genes into CSU program. They're not in varieties yet, but we have material in the pipeline with them in there. I saw something pop up. I don't know if there was something on the chat or not, but.
No, I haven't seen any question.
So we have a question. So Dave Green mentions Fusarium head blight is a problem at times, primarily on high yield irrigated wheat. Yeah, Scott has mentioned that. It's where you do have it under a pivot or something. You have seen Fusarium. And so, something to think about with that. Actually, I can touch on it later, but the timing of that irrigation, if you're putting that water on while that wheat is flowering, especially that kind of 10-day window during pollination, that's you're really increasing your chances of getting Fusarium. Are there known biotypes of, I'm assuming, stripe rust that have led to the breakdown of resistance? Yeah, so like this kind of graph shows, there's a lot of biotypes, a lot of isolates that have, it's just a selection. And so as a variety goes out, and let's say it has YR17 and it's grown on a lot of acres, that mutation is likely already there in the rust population, and it's just a selection mechanism. And once that isolate gets selected for, it's going to increase in numbers, and then it's going to break down the effectiveness of that resistance gene. Great question. Let's try and get back here. My mouse doesn't want to work, but okay. So when I came into Arkansas, there wasn't a plan really for stripe rust. It was kind of haphazard. So we really got an organized plan. The first one was just establishing a really nice screening nursery. And so we would develop spores in the greenhouse. We'd go out with a backpack blower, really when the wheat was pretty small. And, in fact, usually as early as kind of January, we'd go out and hit those with spores. And we do that a number of times. And I'll show a drone image of our stripe rust nursery towards the end of my talk when I talk about drones. And so we kind of did this shuttle where we'd select for stripe rust in 2 different generations. We'd identify new genes, map those, and then we could then use those markers to better select for those new genes. So marker assisted selection. And this is just a really good picture. It's amazing to me every time I see pictures like this, just the how real genetic resistance is, because you can have this stripe rust line here, or this wheat line here, that's nearly completely susceptible to stripe rust, and right next to it is a line that is pretty resistant. Right next to each other, same pressure, but genetic resistance gives you that, that resistance. One thing I kind of had to understand a little better was the idea of adult plant versus seedling resistance. And so a lot of the resistance we have in wheat is adult plant type. And that means you might get stripe rust while that wheat is small, but as it approaches adult plant stage, jointing, it'll actually become resistant. And so we saw that when we kind of started evaluating material at Arkansas. And so we first looked at things in the greenhouse. So we did a greenhouse screening, and these were 2 lines kind of this population we were studying. And we knew that Virginia 96W270 was resistant, and we knew Coker 9835 was susceptible. But when we screened them in the greenhouse, they were both pretty susceptible to stripe rust. And the same was true for our resistant check, which is the old Arkansas variety, completely susceptible, 8s and 9s for infection types, so high infection, as well as for our susceptible check, susceptible. So we knew we were dealing with adult plant type resistance, right? So we take this out to the field, we do a field screening, and then we see our Virginia line being very resistant. We see our susceptible check being very susceptible. And so it's adult plant type resistance. It's very common, and it's in terms of, if you're going to put out a fungicide for stripe rust, the timing of that, thinking about, is it seedling, early, not necessarily seedling, but just early growth stage, or is it getting to jointing, getting, to where it's more of that adult stage where you might have resistance that will kick in. And so it's an important timing aspect. And then we were able to identify 2 important genes in our germplasm there at Arkansas, and it just shows the effect of those. And so we've got on the x-axis here, we've got lines that have no resistance genes. We have lines that have 1, which is located on chromosome 3B, and then we have lines that have a second located on chromosome 4B. And then we have some that have both. And what we saw in terms of disease severity, which was on our y-axis, is if we don't have any of these 2 genes, we have 70% severity on average. If we have 1, we're at kind of 40%. But when we combine those together, we're almost immune to stripe rust. And so this is kind of this, marker assisted breeding put into practice here, where we can select lines that have both of the genes that we want, and we have stripe rust resistance, we can focus on other traits. So that's it for stripe rust. I'm not seeing any other questions pop up in the chat or anything, so I'm going to talk about Fusarium. So another important disease in a lot, many parts of the country. I mean, even in eastern Nebraska, it's an issue. Even parts of Kansas, it's an issue. Luckily, not in Colorado. Obviously, one of the reasons is because we are so dry. So maybe we take a little Fusarium for a little moisture. But, So it's caused by Fusarium graminearum, and it likes, again, this warm, rainy, humid weather, but it's going to infect at flowering. So seedling stage doesn't matter. The growing season really doesn't matter. It comes down to what's happening during flowering, because that's when Fusarium is going to infect. If you're following corn stubble, which is fairly common in Colorado, you're increasing your chances of getting Fusarium head blight, because that corn stubble serves as a host for that inoculum. It causes reductions in yield, grain quality, those kind of things. The big one with this is it also produces vomitoxin, or more specifically deoxynivalenol, or DON, and that is it's toxic to humans, it's toxic to animals, and so there's FDA limits on how much can be present in the grain supply. Similar thing can happen in corn as well. So that's the big issue with this disease. And the hard thing about it is any management needs to be done before you see the symptoms. And so you kind of have to know if you have a moderately susceptible or a susceptible variety, you're going to probably want to do a preemptive fungicide if you know your operation is prone to Fusarium. If you have a moderately resistant variety, well, then there's a forecasting tool you can use to kind of forecast if you need to spray a fungicide or not. Those are kind of two ways to control this disease. So the way we measure this disease in the field, there's a few different ways. Incidence, it's really just how many of the heads in a plot have Fusarium. Generally, we take this kind of a week after heading. Severity, which is how much it's spread in the head. So it infects and it spreads. Generally, it'll infect towards the middle of the wheat head and then spread out. You may go out in an irrigated situation and see it in one little spikelet on the head. And that's fine, but you got to watch it if it starts to spread out from that spikelet. After maturity, we'll harvest the grain and look at Fusarium damaged kernels. This is probably the best way to phenotype for this disease in terms of breeding. So we can see here we've got healthy kernels, 2% infection, all the way up to 75%, where you have a lot of white shrunken kernels. If we zoomed in, you'd actually see some red kernels in there as well, because this the fungus will actually have a red color. So that's Fusarium damaged kernels. And then we also take those Fusarium damaged kernels and we'll send them off to get analyzed for that vomitoxin. And we've, the FDA limit for DON is 2 parts per million. And we've actually seen, as high as 70 parts per million in some lines in our screening nurseries. So to combat this, we focused on introgressing this gene called FHP1, which is, I'd say, one of the most famous and notorious genes in wheat. And it's some yield drag associated with it, but nothing compared to, say, what Scott has had to do bringing in solid stem for sawfly. The yield drag associated with solid stem is much greater than this FHP1 gene. But we worked on bringing this into Arkansas, and really it's interesting if you look, it reduces incidence and severity some, Fusarium damaged kernels more, but the real impact you see is on that DON accumulation. And so it reduces that by about 40%. It's going to prevent dockage. That's going to prevent maybe complete rejection of the grain at the elevator. And then showing how we've incorporated this in at Arkansas. This is some of the last lines I was testing there before I left in the state variety test. And these are two of our kind of standby Czech cultivars, very widely grown in that region. Pioneer variety 26R41, and then Hilliard, which was a public variety developed at Virginia Tech. But in terms of percent of Fusarium damaged kernels, we were doing very well when I left. So we'd incorporated in FHB1 into several lines. And then this AR06 146 was actually the last line I released before I left. So very, very good for Fusarium. So we'd really made a lot of improvements there. You know, from a starting point where, you know, we had kind of low yielding material, we brought in, we increased yield, and when we did that, we brought in some susceptibility to this disease, and so we had to pull it back and balance those two things. And so I think we did a really good job of that. And kind of, I think it was good preparation for working on Sawfly here, because it's figuring out resistance, yield, you know, and all those other traits and how you balance those in a variety. So that's kind of the end of my Arkansas story. If there's any questions before I move on, you could shout those out really quick. I don't see any more in the chat. Unless I hear any, I'll just keep moving. Doing pretty okay on time. So coming to Colorado, you know, it was, this job at Colorado State was just always one where, you know, me and my wife, we told ourselves if it came open and the timing was right, that we would think about it, because it just has such a great legacy of breeders and developing varieties and just the impact, you know, and with the cooperation with the wheat growers and marketing aspect with Plains Gold. It's just a really unique position, you know, at a university. And so, you know, Scott, he loves the fact that he has these pictures going back, you know, with all the former wheat breeders, you know, from Bert Curtis to Jim Welsh, Jim Quick, and then me and Scott, you know, during the pandemic. I mean, I came in and everybody that's working out at the seed lab was wearing masks out there. And so just, you know, we're working through that, but it's been really great coming in and joining this program. So thinking about targets here at CSU, you know, grain yield is always going to be number one, and that includes drought tolerance. That's impacting grain yield. Scott's done an amazing job, you know, with John in the quality lab, working on milling and bread baking quality, really just bringing up the standards in terms of Colorado wheat varieties and how high the standard for baking there has become. Stripe rust, I talked about it. It's an issue, and you know, as we try to incorporate like wheat stem sawfly resistance and wheat streak mosaic virus resistance, it's hard to keep stripe rust resistance in there, and so we have to keep working to get all of that into a package. Wheat stem sawfly, obviously, probably the biggest focus of the program right now. Wheat curl mite and wheat streak mosaic virus, I feel like we're doing pretty good there. And then also, I just added in herbicide tolerance. And so, you know, we have Clearfield, and we have the coaxium system that we focus on in the lab. And so I'll touch on the coaxium side of things. So there's kind of two pipelines to how we develop lines at CSU. And we've called these kind of the conventional, it takes 9 to 12 years to develop a variety, versus the fast track or the 6 to 7 year plan. And so for probably Scott's kind of first 10 years on the job, it was mostly this conventional pipeline, you know, where you're making a cross and all of your advancement is done in the field as you're testing and purifying. And then this fast track pipeline, this involves, you know, making the cross, and you're fast tracking it either through our doubled haploid program, which is kind of a way of tricking wheat, using corn pollen to trick wheat into thinking it's pollinated. And then you can develop an inbred line very quickly. Or using just a quick greenhouse cycle where you're cycling things through really fast. And if you talk to Scott, I mean, he'll tell you most of his really good stuff recently has come out of this fast track pipeline, and particularly the doubled haploid pipeline. So it's really nice to have that in-house. I'm not going to talk about those methods today, but no. And then the big, silver bullet of his program is the testing. And so it's, we test on up to 15 locations, cooperating with growers in the state. And it's been great going out. I was involved in planting and, just getting out and into Eastern Colorado, and I look forward to doing more of that. But the testing aspect is just the why his program has been so successful. And there's crossing over between these. So parents will cross over once things come out of the end of the pipeline. With the fast track, you're talking 6 to 7 years instead of 9 to 12. But you take those, you put them back into the crossing. Whether it's conventional or fast track, you get them back in as quickly as possible. You try to close that loop. So I chose to talk about coaxium a little bit, one, to kind of educate myself on it, and another is we just released a new line for this system. And so this coaxium production system, it's basically a way to control grasses in a grass, right? Wheat's a grass, and you can control ryegrass, goat grass, and other grasses in the system. And so the coaxium, it combines a patented herbicide trait known as Axygen. It's non-GMO, which is the beauty of it. Similar to Clearfield, it was developed using a mutation breeding approach. And so you are tolerant to Quizaloflop, the group 1 herbicide. And so this trait has been introgressed into CSU germplasm as well as other programs. It's owned by the wheat growers of Colorado, and it's licensed out to other programs. And then you combine that with Aggressor, which is the herbicide that's tied to this. And so, it's a tool system where if you have a grass problem, you can use this as a tool to clean up that problem. It's not something, there's a stewardship involved there where you have to rotate the system with conventional or Clearfield. But it is a very, very nice tool to be able to control these grasses where we need to control them. So Incline AX was released by CSU in 2017, and it was really the first widely available line for the coaxium system. And in our program, we developed these doing, few different methods. It could be conventional, fast track, or using kind of a back cross approach. Almost always, we're going to be using marker-assisted breeding to help us detect whether we have the trait or not. And then we have field screening at the preliminary stage, a qualifying trial before a line is released, where you have to have 6 locations of data. And then we use the same extensive testing we do for other lines. So testing for grain yield, testing for other important traits. So I thank Todd Gaines for this slide, but the Axygen trait, it was developed, like I said, through mutation breeding. And basically the result of one base pair change in wheat, where you had a change in amino acids from an alanine to a valine. And so wheat, it has 16 billion DNA base pairs. It's huge, but a change in one of those resulted in this herbicide tolerance. And there's actually 3 different mutations, one on each of the wheat has 3 ancestor species. And so we actually have a mutation on each of those ancestor species genomes. And as you have 1 versus 2 versus 3 mutations, you can actually build up the resistance to be able to use higher rates of this herbicide. This is what it would look like in the field. This is the wild type A allele there, but here we've been able, these have the mutation, so we've been able to spray these with aggressor and not kill them. And so it's a really fantastic tool. Marker assisted breeding, you know, it's a pretty simple process. We take tissue samples of the lines we want to test. We isolate DNA from those. We do what would be called a polymerase chain reaction, or PCR, and then we score those to determine if we have the wild type or the mutant allele. So this is an example of what it would look like to determine what allele we have. And so here we have mutant alleles, so these would all be tolerant to the herbicide. We have lines that have one of each allele, heterozygotes, and then we have a handful of wild type alleles. And so we do this as early as we can to try to weed out those lines that don't have the herbicide resistance before we actually take them, you know, to the field to spray them. So here's just an example of what the data would look like. Crescent AX, it has mutant type at the A and D ancestor genomes. And then we've got some breeding lines here. And most of the lines we have A and D herbicide tolerance. That gives you a little better, a little less injury compared to something that has the B version. But we are working to develop 3 gene type coaxium lines as well. And I'll talk about that a little bit. It's just a couple of pictures of what would look like in the field. I showed you one of the wheat just completely dead, but here you can see some injury in the qualifying trial. And then here's a greenhouse screening that we used last year, working with Eric Westra here at CSU. And so here you have bird, which is obviously susceptible, it's dead. Here's incline, maybe showing a little more injury than our new release, which is the Cavari AX. So just some, you know, you can get some level of injury with this herbicide, depending on the variety. Some varieties show a little bit more injury. You know, generally, even if you had this type of injury, you might grow out of it and not see a yield penalty. But really, our goal is to avoid any kind of injury at all. Computer's being a little funny. Okay. So coaxium varieties for Colorado. Crescent was released a couple of years ago, and then we followed that up with Cavari AX this year. And so pretty similar in terms of agronomics. There are a few differences between these two lines. If you look at just some head-to-head data of these lines. So here I've got, you know, traits of interest, and then I have Cavari and Crescent. And just a couple things of note, Crescent tends to be a little bit early. It has gotten nicked from freeze, I know, a couple of times in Colorado. Cavari is a couple days later, so than Crescent. And then from a yield perspective, Cavari has just had great yield potential, even compared to Crescent, which is pretty good. But just looking at our head-to-head data, it's a 5 bushel advantage over Crescent. One thing I will say also about Cavari is that in a high yield situation, it does tend to lodge, and lodge is a little bit more than Crescent does. But in general, higher yield potential, a little bit later maturity than Crescent. And then I mentioned the 3 gene aspect. And so, you know, looking here, this is just some pictures of controlling goat grass. And so, you know, untreated, it's growing pretty, there's no weed here, it's just goat grass. You know, 10 ounces of aggressor, 16 ounces of aggressor, controls it pretty well. You can, I believe you can actually do up to 24 in different combinations. But, you know, the idea of a 3 gene coaxium, currently there's only 2 gene coaxiums on the market. would really give some more options for chemical, different chemical rates. So we've got some really nice 3 genes in the pipeline. And so just looking at a couple here, grain yield across multiple locations, outperforming Crescent and actually outperforming Guardian, which has been really high yielding the last few years. So, hoping to get those out with, it won't be next year or the year after that, but in the next few years, getting a 3 gene coaxium out. And finally, the last kind of thing, I think I'm doing okay on time, Wilma. If I'm not, please let me know. But just looking at what we've been doing in terms of sawfly. And so this is a new, I've never really had an insect pest that I've worked on as much as I'll be working on this pest. And so we think about kind of the development of sawfly. It's been a problem in the spring wheat region, even going back to the early 1900s. 1980s kind of moved into Montana Montana winter wheat. 2000s into Wyoming, and then 2011 into Colorado winter wheat. It causes a number of things, inhibits translocation of photosynthates to the grain to fill those grains out, which is going to reduce yield and it's going to reduce test weight. And then we all know it cuts the stem, makes it very difficult to harvest. And then you're not going to have as much residue left after the season either. Management is very tough. So insecticides are not effective. Parasitoids, we're still kind of learning about those. Cropping systems may be partially effective. And then we have variety resistance, which we've been working on, which has shown at least partially, be partially effective. And I'll explore that a little more. Just thinking about the life cycle of this pest, it's going to emerge, from last year's stubble. This is going to be May or early June, and it's going to lay that egg in the wheat stem around the same time as head emergence. You're going to get larval development, and then it's going to create that stub and the notch, and which is going to end up causing that lodging, and then you're just going to repeat that cycle year in and year out. And that's, one of the reasons that this problem is not going away is that you get, you have last year's stubble, you've got fly just waiting there to come back the next year. So it's kind of it's a tough cycle. So the most impacted areas are still going to be in the northeastern counties of the state, but it has moved down more or less to every wheat growing county in Colorado. And so, there's a survey that's done every year. The red indicates greater than 50% of the field surveyed had infestation. And so it's just, slowly moving down. Maybe I'm not going to say even slowly, it's moving down the state. So back in 2011, Scott started working on this problem. He evaluated solid stem varieties from Montana State, started making crosses using doubled haploid and marker-assisted breeding to really attack this trait. And now we're doing a lot more just conventional breeding as well, selected bulk approach, and then also looking at some non-solid stem-based resistance that could be combined with that solid stem. So the number of crosses, this shows time on the x-axis and the number of crosses that the program has made on the y-axis. So first crosses made in 2011, 2012. There were then a big group of bird derivatives from these that incorporated in that solid stem trait, and then a group of Antero derivatives after that. It's kind of been the progression of just ramping up our breeding for sawfly resistance. Fortify SF was released maybe a year ago, but really just, is out there really this year. And so pretty good agronomic package. I mean, it's not solid stem, it's semi-solid stem. I'll talk about that in a little bit more. But it has test weight, good baking qualities, and good yield in a soft lie situation. It's not going to be comparable to our other conventional hard reds in a non-soft lie scenario, but in that where you are going to get cutting, it's done very well. The stem solidness, you know, it's done on a scale of 1 to 5, and then at 5 different sections of the stem. And so the highest it can be is 25. So it ranges from a completely hollow 0 to a completely solid 25. And it's basically going to increase larval mortality. It's harder for that larva to chew the stem. It explains about 75% of the phenotypic variation for solid stem. This one locus does, excuse me, this one gene. So there's still some other things out there that we can do. But one of the tough things, it is influenced by the environment. And so in some locations, you'll have better expression of this solid stem than in other locations. And there's a couple of different versions of this solid stem. One is called the rescue allele, which is just present all the time. So I'm calling it non-temporal. And then this Conan allele, which is present early in the season, and then is supposed to kind of, the stem hollows out as the season goes on. And so there's a kind of a question, can this temporal expression be more favorable for yield? And so we do see a yield penalty with solid stems. So if that solid stem kind of fades at the end of the season, can that help have higher yielding wheat? And so we actually looked at this a little bit last year. And I'm going to show a little bit of data. This is from Montana first, and then from Colorado. But we have a couple different versions of lines here. And so this is Yellowstone. It's a hollow stem line. Montana State incorporated in this temporal expressing solid stem. So it's expressed early, and then it kind of fades. So measured solidness twice. And so early, it had a solid stem rating of around 16. When they measured it later, the solid stem had decreased. So it lost about 7 points. And that was it. So that's the Conan. In just our hollow stem, there wasn't really much of a change. And then this set of lines here compares the temporal, that Conan, to just your regular solid stem in a different background. And so when they looked at this in a different background, you did not see that fading away of the solid stem like we saw in that Yellowstone background. So there's a little bit of genetic background going on with this trait. And then again, we actually saw an increase in solid stem in our just regular solid stem variety. And then Warhorse is kind of a Montana, just really solid stem, really good variety up there for them. So really high, 20 out of 25 solid stem rating. We looked at these same lines in Colorado, and the trend was there, you know, for Yellowstone, magnitude was much less. And so it was a 14 early and kind of faded to 12. The hollow stem, you know, didn't really do a lot. And then we saw the similar result for Judy, where we didn't see fading as much of the hollow stem. And then I threw in Fortify SF as an example, where it's a semi-solid stem, so it's usually around a 12 for its solidness, and it maintains that throughout the season. 5 minutes left. Okay. Ugh. Every time I click on that chat, it gives me a little problem. So kind of the last couple of slides here. So we also looked at yield of these lines. This is at Orchard and then at New Raymer. And then here's grain yield here. And so Orchard, we really didn't see any effect on yield for that temporal expression versus the regular solid stem. At New Raymer, we actually did see some effect on yield. Granted, it was a low yielding location, but this blue bar here where we had temporal expression, so where that solid stem faded as the year went on, we actually saw it being about 5 to 6 bushels higher than just our regular hollow stem here, and then higher than our rescue allele as well in Judy. Okay. So last couple slides on softfly, we have lines that we're increasing this year in Yuma, Arizona, and trying to build on that success of Fortify. So if we look at just head to head, this is 16 SF070 versus Fortify. It's going to have a more solid stem. So like I said, Fortify is that 12, around 12 solid stem rating. This is going to be closer to 15. From a cutting perspective, they actually have similar cutting, not significantly different from each other. Yield-wise, this 070 line, we hope can be a little bit higher yielding than Fortify. So on average, it's around 5 bushels higher. And it actually does better than other solid stem lines in a non-softfly, under non-softfly pressure. The second one is this SF067. And so again, it has a more solid stem than Fortify does. Similar cutting, at least in our nurseries. Yield-wise, it's not significantly different. Numerically, it's a little bit lower yielding, but we really think this more solid stem will be beneficial when it gets out on a grower's field. And then last slide about softfly, we've kind of done some shifting in the program. And part of that is we're now using Akron as kind of our main softfly site. And we've really ramped up what we're doing there. So we've got about 20,000 head rows we're growing there this year. We've got about 500 just lines that we're testing. And then we also have a bunch of bulk populations. These are earlier generation populations that we're hoping we can select under softfly pressure and get some good selection for that solid stem trait. Then just thinking about how these are going to perform, we have the ability now to predict how things are going to perform. And this red line here in the middle of this graph represents new lines that we haven't actually tested yet. But compared to how our lines have performed in the past, we should have a bunch of lines with lower stem cutting compared to anything we've had before. And so we're really excited about what could be coming out of this nursery. So if I've got a minute, I'll just finish up. One thing we're going to work on moving forward is some high throughput phenotyping. I did some of this at Arkansas, and this is a drone footage I mentioned of our stripe rust nursery. And so we worked on doing some high throughput phenotyping for stripe rust severity. So we took images of our stripe rust nursery. We analyzed them for color. You can see here a lot of stripe rust infection and kind of mixed in within the population. But we were actually able to really predict really well stripe rust severity in this scenario. And so up to an accuracy of 0.73 to 0.85, depending on our plot size. And so I think there's a lot of applications for doing this drone work in the future, not just related to disease phenotyping, but also estimating biomass and really trying to focus on drought tolerance using kind of thermal imaging. And we're doing this in collaboration with the Drone Center at CSU. And so I'm going to stop there. I'll thank my current lab, or my previous lab at the University of Arkansas, as well as the lab here at CSU, Eric Westra, Todd Gain for sharing data and slides with me. And I'll be happy to take any of your questions.